Welcome. I'm Kathleen Seib, Vice President of Research at Ohana Biosciences. Thanks for joining me for this virtual presentation of our research on developing a novel sperm preparation procedure that increases human sperm hyperactivation, which I'm presenting on behalf of my colleagues and co-authors, Shannon Rainsford, Joe Broncali, and Felipe Navarrete. Before diving into details of this project, though, I'd like to take a moment to introduce Ohana Biosciences. Ohana is a sperm biology company dedicated to developing new technologies and therapies targeting sperm function and reproduction. We've built a sperm biology platform where we have applied cutting-edge single-cell sperm analysis combined with sophisticated computational biology and machine learning to develop multidimensional profiling of individual sperm cells. These data have led to novel insights into sperm function and heterogeneity that form the foundation for developing products to improve sperm function in assisted reproduction, to block sperm function as non-hormonal contraceptive for both men and women, and new technologies to reduce sperm-mediated risks of pregnancy complications and diseases in children. This presentation is focused on one of our fertility programs. Infertility affects 7 million couples in the US, and 90% of those couples are not treated, primarily due to the financial, physical, and emotional burden associated with assisted reproductive treatments. In the past 4 decades, there's been a 50% decline in sperm counts, along with reductions in sperm quality. However, innovation in infertility has been limited and primarily focused on treating the female. Our view is that by improving sperm function in assisted reproduction, we have an opportunity to increase effectiveness of the treatment and reduce the number of cycles that are needed to have a baby. For couples who need to undergo in vitro fertilization to have a baby, it's a daunting treatment with pretty limited success rates. Even with a high-quality embryo, only about 50% of embryo transfers result in a baby. In the US, each cycle costs on average $16,000, with only a 20 to 45% chance of success, and most patients will do 2 to 3 cycles. Much of the focus in assisted reproduction has been on improving the number and quality of eggs that are available and on in vitro embryo development. The way sperm is prepared for IVF and IUI has not significantly changed since the introduction of density gradient centrifugation in the late 1980s. By improving sperm function in fertilization and early embryo development, we have the potential to increase the success rates of each cycle and reduce the number of cycles needed to have a baby. So what are we doing that's different? It's well understood that sperm need to be activated in order to fertilize the egg. Most media for sperm preparation today are based on the composition of human tubal fluid, which sperm encounter as they get close to the egg during natural conception. However, during natural conception, sperm undergoes several stages of capacitation as they travel through the female reproductive tract before the final stage of capacitation when sperm comes in close proximity to the egg. Sperm are deposited into the vagina, and motile sperm penetrate the cervical mucus to get into the uterus. Once there, sperm are triggered to swim fast and straight across the uterus with progressive motility. As sperm approach the fallopian tubes, they undergo another stage of capacitation that's associated with an intermediate or head-wagging motility. And finally, as sperm come in very close proximity to the egg, it undergoes the final stage of capacitation, adopting a hyperactive motility pattern and undergoing acrosome reaction. Only fully capacitated sperm can penetrate the cumulus cells surrounding the egg and bind to and penetrate the zona pellucida to fertilize. What we've done is developed a sperm treatment that is designed to mimic this stepwise activation that occurs during natural conception to improve sperm function in IUI and IVF. Ohana's sperm treatment is a clinical stage product for preparing sperm for IUI or IVF. Sperm are isolated from seminal fluid, and then critical capacitation triggers are added sequentially to activate sperm. For IVF, sperm are fully activated by sequential addition of 2 capacitation triggers, and then can be used for insemination, or it can be pelleted and resuspended in a preferred fertilization media. For IUI, 1 capacitation trigger is added, and sperm is ready for insemination, as they will encounter the 2nd capacitation trigger in the oviduct. Following this stepwise activation, the percentage of sperm that are fully capacitated, as measured by a hyperactive motility pattern, is increased relative to standard methods for activating sperm ex vivo. The commercial product based on this research will be called spertility. In a mouse model of IVF, the Ohana sperm treatment significantly increases sperm hyperactivation, fertilization rate, and day 3.5 blastocyst development from the total number of oocytes that are retrieved, compared to a TYH buffer, which is a standard buffer for activating mouse sperm. Additionally, we observed a 1.5-fold increase in the number of blastocysts that develop from 2-cell embryos and higher live birth rates with the Ohana sperm treatment relative to control, indicating that the effects of the sperm treatment may have impacts even beyond fertilization. In a mouse model of IUI, we observed significant increases with pregnancy rates and live birth rates in females inseminated with sperm processed using the Ohana sperm treatment compared to control. We saw a 3-fold increase in pregnancy rates and a 5-fold increase in live birth. Taken as a whole, these data suggest that activating sperm using the Ohana sperm treatment improves sperm function in fertilization and embryo development, increasing pregnancy and live birth in both IUI and IVF in mice. Building on these learnings from mouse sperm activation, we developed a sperm treatment optimized for human sperm activation. In the data shown here, sperm samples from men and couples seeking treatment for infertility were split and processed in parallel using the Ohana sperm treatment and a commercially available sperm wash as control. Sperm motility data was collected by computer-aided sperm analysis, or CASA, and analyzed by CASANOVA. In these samples, there's a decrease in the sperm categorized to slow and a corresponding increase in sperm with progressive, intermediate, and hyperactive motility patterns compared to control. When we focus on one parameter of CASA called curvilinear velocity, or VCL, which is increased in sperm that are classified as hyperactive, we can plot the data as a distribution of individual sperm cells. These data show that when sperm are processed using control sperm wash, as shown in orange, the distribution of VCL in sperm from men seeking treatment for infertility is lower than in sperm from healthy donors, shown in blue. When sperm is processed using Ohana IVF, the distribution of VCLs from men seeking treatment for infertility in red is similar to that of healthy donors, again shown in blue. And even in healthy donors, Ohana IVF shifts the distribution of VCLs higher, shown here in green, indicating that the stepwise activation of sperm improves sperm motility in infertile or subfertile men, as well as in normally fertile men. These results show the full change in percent hyperactive and intermediate sperm in individual sperm samples processed with Ohana IVF relative to control. In these samples, 90% had at least a 25% increase when prepared with Ohana IVF relative to control, indicating that this treatment improves sperm motility in most samples tested. While we were optimizing the Ohana sperm treatment for human sperm hyperactivation, we were also reviewing the literature to understand the correlation of sperm hyperactivation with IVF success in humans. In this study, shown here on the right, they used CASA analysis of sperm samples from 150 couples undergoing IVF and showed that there was a 2-fold increase from 5% to 9% in median percent hyperactive sperm in couples that achieved pregnancy relative to those that didn't. When we analyzed the CASA data we generated using the same definition of hyperactive as in the publication, sperm processed using control conditions had 7.5% hyperactivation compared to 17.5% in sperm processed using Ohana IVF, a percent hyperactivation that was strongly associated with pregnancy in the published study. However, a correlation in the literature is not definitive, and we are very interested to see if the effects of fertilization rate and embryo development that we observed in mouse IVF translate into humans. We've designed a clinical study to compare Ohana's sperm treatment to a standard commercially available sperm wash in a split insemination design where we're monitoring for fertilization rate, embryo development, and the number of high-quality euploid embryos available for transfer. In summary, Ohana has developed a novel sperm preparation procedure designed to mimic how sperm are activated during natural conception. We've shown that this process improves fertilization, blastocyst development from fertilized egg, and live birth rate in a mouse IVF model. In the human sperm samples tested, Ohana sperm treatment restored normal motility parameters in sperm from subfertile men and improves motility in sperm from healthy men. The observed improvement in percent hyperactivation associated with Ohana preparation is associated with pregnancy success based on published data, and we're conducting a clinical trial to evaluate the impact of improved sperm function with the Ohana sperm treatment on fertilization rates and quality of embryos. Building on this data, we'll be developing our commercial product called Spertility. Thank you.